What's going on, folks? How's it going? Look like we got several people in the room. Awesome. Good stuff. I was actually watching my good friend Jeremy Krug in his stream a little bit. I knew absolutely nothing as to what was going on, but yeah, it looked like he was doing some fun stuff. All right, so what my plans are for this evening. Last night we talked about Unit 3, which is cellular respiration, photosynthesis, enzymes. We talked about most of Unit 6. We went through the 2024 FRQs together, which is sort of a content sort of review, because you're going through a lot of content. Yeah, Krug is a goat for sure, 100%. It's one of my great life privileges working side by side with him in the same building. So it's pretty crazy. So we're gonna go over tonight. I want to look at some Unit 5 stuff. I'm not getting too deep in the weeds with Unit 5, just because there's not a lot of Unit 5 on the exam. And I want to go over the second part of Unit 6. And then I want to look at the FRQs from 2021. And then if there's anything else that you guys have questions over, I started with this particular slide just to show you sort of the weight of the different units, right? So you can see that Unit 5 is not heavily weighted. At most, it's going to be 11% of the exam, which is not a lot. It's not enough to fail you. And my students really struggle with Unit 5. And Unit 6 is going to be pretty heavy. Unit 7 and Unit 3 and 6 are the heaviest units. And so I haven't spent a lot of time on Unit 7 because it's not too difficult. And it's one of the last units we did. And so I didn't spend a lot of time going over it last night. And I probably won't tonight. But it kind of finds its way into all of the questions. So without further ado, we will go over Unit 5. And do definitely recommend getting some sleep tonight, but if you're up, I have videos of all these units where I go through, basically teach these slides and walk through them. So if you're up and you will just want to kind of rehash some vocab in your mind, definitely a good use of your time to do that. I think that is probably one of the best things you can do, is just sort of rehash some of the terminology so that you're not surprised by anything on the exam tomorrow. So let's look at Unit 5. First, let's look at this process, meiosis. It is a process that produces haploid gametes from diploid cells, and it produces 4 genetically different haploids. The concept of haploid and diploid is very important. Haploid, let's talk about diploid first, actually. A diploid cell represents a cell in which the individual has 2 of each copy of every chromosome, right? So chromosome 1, there's 2, chromosome 2, there's 2, chromosome 3, there's 2. Right, and where did you get 2 each of these? You get 1 from mom and 1 from dad. This makes you a diploid organism. So when the organism goes through meiosis, the sex cells go through meiosis, they create haploids, which are half of these, right? Each pair, which are called homologous pairs, is going to choose, not really choose, but you get the idea, it's going to choose one of these chromosomes. And so for a human genome, there's 23 pairs, and so there's 23 separate choices, right? So it creates a lot of different combinations. And so that's haploid and diploid. Diploid's always 2N, haploid is N. And it is a math problem. If 2N equals 8, N equals 4, not 16. I feel the need to say that every time. When I teach my students meiosis, I do not teach all the phases. I just teach interphase. Interphase is important because that is when DNA is replicated. And you have to replicate the DNA so that you can go from 4 to 4 ones, right? Or from 2, so that you can go from 1 2N to 4 Ns, right? You have to replicate it first. And I teach prophase 1. Prophase 1 is where crossing over occurs, and so that's an important phase. And I teach anaphase 1 because anaphase is where homologous pairs separate. The ones that you got from mom separate from the ones you got from dad, up all the way down the cell. And then I teach anaphase 2, and this is where sister chromatids separate. Sister chromatids is like, this is really weird looking, but it looks like some like cheap, like 70s countertop or something. So if this is a chromosome that has replicated, right, there's 2 of them now, and then sister chromatids separate, and you do that, right? Okay. I have some pipe cleaners on my board in my classroom, and I use those, and it's pretty helpful. But yeah. Yes, I will do that. That will be a helpful exercise for sure. So just remember with meiosis, mitosis, the difference, I think almost every year I see something. One of these 2 is going to sneak into the FRQ as sort of a, what is the products of it? What happens how does meiosis look if this happens, or something along those lines? All right, so I think this is real helpful. Mitosis, you always end up with 2 diploid cells that are identical to one another. Meiosis, you end up with 4 haploid cells that are not identical. Why aren't they identical? Has to do with the choices that are made to appear. We're going to give that a name in a minute, and it has to do with crossing over, which is what we're talking about here. Crossing over is just trading spots of homologous pieces of chromosomes, right? How can it trade? It's sort of like, you know how your left and right shoes are not identical, but they're homologous. If you took like one of the little lace holes, they probably have a technical name, off of your right shoe and put it on your left shoe, they're not identical, but it would work, right? And so that's why these 2 pieces can trade. They're not identical, but they're the same version, or they're the same genes, just different versions of those genes, or different alleles, right? Independent assortment, this is the choices that we were talking about earlier. So if if purple went this way, whoops, I don't guess we're drawing today. There we go. If purple goes this way and pink goes this way, which way does blue go? Does it go with purple or pink? Well, it can go with either one, right? Which way does dark green go? It can go with either one. And so that go with either one is independent assortment. And this has to do with how you set up dihybrid crosses, right? Which we will look at in a moment. Actually, let's look at it right now. So here's an individual that is heterozygous for Y and heterozygous for R, right? They have big R, little r, big Y, little y. And so their genotype is going to be, there it is, it's down there. All right, here we go. Their genotype is going to be this, right? And so in order to set up a dihybrid cross, so let's say you have this, right? If you have this particular cross, the way that you set this up is you just decide how many different combinations. Actually, let's change it a little bit. Let's do that. How many different combinations between R and Y can you have? Or in other words, how many different combinations between pink, purple, and blue, light blue can you have, right? It's the same thing. And then you do like a big R, big Y, big R, little y, little r, big Y, little r, little y, right? And for this one, it's different. It's less, right? It's big R, big Y, and then big R, little y. That's it. And so then you set up a square. But here's the thing. I've never seen In AP, I've never seen you have to set up a square like this. And at most, they're going to ask you to know the ratios in a monohybrid cross, something like this, right? Which is just something that you can sort of figure out in your head. It's going to be like 1/4 that, 1/4 this, and 1/2 this, right? And then the only one that I've seen that expected to know, and I've only seen it once, is just when it's dihybrid for both, or when it's heterozygous for both traits, both organisms, and it's a 9-3-3-1 ratio, where the 9 is dominant, the 3s are dominant recessive dominant, and then the 1 is recessive. Other than that, I don't expect you to see that. Yes, I will go through the sex-linked thing. And are genes passed down through mitochondrial DNA considered sex-linked? No, because a sex-linked gene is linked to the sex chromosomes. Mitochondrial DNA is considered non-nuclear DNA, which we will look at in just a moment. Good question. So the only type of question that I expect you to have to know the ratios on would be like a chi-square problem. And frankly, if it's a chi-square problem in the multiple choice, if it's towards the end of the exam and you know you have time, sit down and work it. If it's at the beginning of the exam, my advice would be to skip it and go back to it later, because you don't want to get bogged down on a math problem that's going to give you 1 point. So if it's in an FRQ and you have time, maybe solve it, particularly if the answer to the question is going to cause you to get other questions right. And I think that is helpful. But in general, I think parking on a chi-square problem is not a good test-taking skill unless you can do it quickly, right? But for a lot of students, it's abstract and it's hard. Yeah, I will go over chi-square. I'll do that. I don't think you'll have to know a lot about the different kinds of cells at all. And any kind of cell that you have to know about, you'll likely be given that information in the prompt, because we're not asked to teach that to you guys. So a lot of times teachers go overboard with like specific information, and we're just not asked to give a whole lot of specifics in the course description, and any sort of specifics you're typically given in the FRQ prompts. And I'll show you that as we're going over FRQs tonight. I think that'll be helpful. A lot of times students kind of get scared about specifics when you just have to know generalities more so. Yep, I can look at cladograms as well, for show. All right, so I don't know if that answered your questions about the dihybrid thing. There's not going to be a time where you have to set up a full dihybrid cross. Each of the 4 structures, are you talking about macromolecules? Yeah, so macromolecules are easy. Carbohydrates only have CHO in them, and C is always equal to O. Lipids have CHO also, but there's a whole lot more CH than there is O. Proteins have nitrogen in them, and nucleic acids have nitrogen and phosphorus in them. That's the easiest way to remember it. And then the proteins always have the R group. The amino acid has the little R group thing that's usually says R, or it's some sort of big thing. Yeah, primary, secondary, tertiary, quaternary. Yeah, so those are just protein shapes. Primary is just a chain of amino acids. You know what? I don't have unit 1 here. It's so weird. Maybe it's here. Yeah, here we go. Primary is just a chain of amino acids. I always call them Mardi Gras beads in my class. Secondary is alpha helix, beta sheets, and they're connected with hydrogen bonds. And tertiary protein is connected with disulfide bridges, is what you'll see commonly. But the tertiary structure of a protein is typically a completed protein. Some proteins are quaternary, and they'll have multiple tertiary structures combined. Not too bad. Okay, moving on. Fertilization, nothing new there. Okay, no. So yeah, there's a lot made of this probability stuff, and no, I just don't think it's helpful. I do not think it's helpful because you might have 2 problems and you'll get bogged in it. I would rather you get all the easy ones right. And so what I teach my students is if you have, I can do Hardy-Weinberg. There's typically, and we're getting ready to do a pedigree. There's typically 1 or 2 Hardy-Weinberg problems, and I've been doing this for 13 years, and I don't remember seeing a population math or Simpson's diversity or any of that stuff on an exam. So I don't expect them to start now, but they might. You never know. It's on the formula sheet, so you might have to do it. So yeah, the mapped unit thing, I would think that, I have seen the linked genes thing, and it usually shows up either in, it shows up in one of those big prompts where it's for the next 5 questions, use the following data, and it'll show 4 different versions of the offspring. And then you have expected outcomes, and then you have observed outcomes, and then it might ask you, why are they so different from the expected outcomes? And you could say linked genes, and the reason that you would say linked genes is because they are linked together less than 50% of the time, or more than 50% of the time, or whatever. But that's the only way I've seen it. I've not seen it over and over. You might see it 2 questions, so I wouldn't lose sleep over something that's specific. I just think it's really edge case. So I know when you're studying for Unit 5, it's important, right? But when you're studying for Unit 1 through 8, it's a whole lot less important. So if that helps, speaking to one of my students, when we went over Unit 5, the possibility of seeing on the test was pretty high, right? But when you go over Units 1 through 8, the possibility of seeing on the test is not as high. That's the best way to say it. All right, so let me look this. I'm just going to do this probability thing real quick, right? So if I was asked to solve the probability, what is the chance that this organism right here would be born to this? How would I solve that? Well, I'm going to do the y's first, right? What is the chance that little y will come from this pairing? And that's 1/4, right? So 1/4, that's a big pencil. All right. And then what's the chance that, this is going to come from that pairing, and it's 1/2, right? Put my X out of highlighter there. Oh my gosh. And so what is the chance that this organism is going to be seen? It's 1/8. I'm left-handed, sorry, and I'm trying to write with my right hand. So there's that. Okay, so that's how you do that probability. That could make a good multiple choice question, right? That shouldn't be hard. There's linked genes. I'm not going to spend a lot of time talking about it. Let's talk about sex-linked real quick. Actually, okay, so sex-linked, first of all, not every gene is sex-linked. It'll say in the prompt, such and such is a sex-linked trait. And it may even give you a little more information, right? Like there was one about fruit flies, and it says it's a sex-linked, and fruit flies have XY genes. If it's about birds, it'll be XY, and the Y is the gene that has everything on it, and the X is the determining gene. So it'll tell you that information. You don't have to memorize all the animals' sex-linked patterns. That information will be given. But when you see the word sex-linked, what you know is that you have genes that are only being carried on sex chromosomes, right? And so if it's an X-linked gene, then it's not being carried on the Y chromosome, right? It's not being carried on the Y chromosome. And so everything's the same, except for you just have to write the X's and Y's down because it's being tracked on those particular things. And so you might have your box, this is a mental box. You can draw, well, actually you guys are going to be doing like Chromebooks or whatever. I don't know if you're getting scrap paper or not, but you could draw on something. How do linked genes and recombination frequencies help us map chromosomes? The distance between two genes is going to be their crossover frequency. So just to answer it real quickly, I've not ever seen it on an FRQ. I've seen it in multiple choice questions, typically in the big prompt questions, like for the next 4 questions, use this data, that sort of thing. I've not seen it on an FRQ. I could be wrong, but I don't remember. I don't remember things from one day to the next. So B and C cross over 9% of the time, and that's how far they are apart. CN and VG cross over 11% of the time, and so B and VG cross over 20% of the time. So yeah, you can take a TI-84 in there. For sure. Okay, so I'm going to do this on every sex-linked problem, right? That looks like an X2, sorry. Yeah, you will need it unless you can do square roots in your head. If you can, man, bravo. Well, actually, a lot of the square roots you can do in your head. So you'll write and then you track your genes on there. So how do you do a pedigree for this? Here's a great one. So this is telling us it's an X-linked recessive pedigree, but how would I know if I just like sort of came to this, right? How would I know? Well, let's look at it. So let's look at some patterns. First, we have mom and dad up here. Mom has it, dad doesn't. Look at their 4 children. All of their sons have it. None of their daughters do. Okay? And this is interesting. And here, everybody has it because everybody has it up here, right? And here, nobody has it. But yeah, you need to bring a pen and a pencil. Why your school would tell you that. Schools are weird. So what this is telling me, if only the males have it, it's because mom gives the X chromosome to their sons, also gives it to dads, but dad has a negative negative, what am I saying, negative, has a dominant allele on his X chromosome, and he gives X to daughters, and so these daughters are heterozygous for the trait. This male doesn't have it, and so this male just has a one big A, right? This female has a big A, little a. And so there's no way that the daughters are going to have it. No way that the daughters are going to have it because dad gives them a big A. And the males can have it because mom has a chance of passing the little a. And so this would be a sex-linked pedigree. Sorry. What's the difference between incomplete dominance, codominance, polygenic? Okay, look at the words. Just look at the words themselves. Incomplete, so it's not complete. It's not completely dominant, right? So it's not completely dominant. So if you have red as dominant and white as dominant, and neither one of them are completely dominant, what are you going to get? You're going to get pink, right? It's a blend. What about codominance? Think about the word cooperate. If you cooperate with someone, what are you doing? You're sharing the operation. So codominance shares dominance. So if it's red dominant, white dominant, then that would be red-white. Polygenic literally means more than one gene. Right, poly, more than one, gene, more than one gene for a trait. And so this is like when one trait, say like height, has lots of genes. So to answer Kevin S., to answer your question, that would be that'd be my first tip off. I would definitely go down to the next level and make sure that same assumption follows out in the next level, right? And so that's what you're doing, is you're essentially eliminating why it can't be this, why it can't be that, right? And so that's how you do that. Okazaki fragments form because the strand is not being continuously replicated. Yeah. Oh, somebody else said that. Beat me to it. Prohibited items are pens and pencils. Man, I'm so thankful I go to like a school that makes sense. Let's see. There's polygenic right there. How do errors like non-disjunction? So look at the word non-disjunction. What is the word? What does the word non-disjunction mean? So first of all, look at junction. What is a junction? Where two things join, right? Disjunction is not where they join, it's where they separate. So non-disjunction is the failure to separate. And so if they don't separate, that means that some cells are going to have more chromosomes than others, and some cells are going to have less chromosomes than others, right? Man, getting bogged in the chat here. That's good. I like all the questions. Thank y'all for asking lots of questions. Yeah, I can go quickly over that. So someone earlier asked about mitochondrial inheritance. The way that you can know about mitochondrial inheritance is if the mom has it, all of her children will have it. If she doesn't have it, none of them will. So mitochondrial inheritance is passed through the mother. I think I hid another pedigree over here someplace. Where did I hide it? I thought I hid it. Maybe I didn't. There it is. Come back. Okay. So here's another pedigree that we can look at. Yeah, non-disjunction +1 -1. There's going to be 4, right? If non-disjunction occurs in anaphase 1, then there's going to be 2 pluses and 2 minuses. If it happens in anaphase 2, there's going to be just 1 of each, and the other 2 are going to be normal. So here is an autosomal recessive. Here's an autosomal recessive one. So how do you know it's autosomal recessive? Well, mom and dad don't have it. One of their sons has it. That checks out. What about here? Why couldn't it be sex-linked recessive? Well, in order for this daughter to have it, then dad would have to have it, right? Because he gives one X away. And so this is autosomal recessive, not sex-linked recessive, because if it was sex-linked recessive, dad would have to have it. He doesn't, so it's an autosomal recessive. Yeah, Autosomal. Yeah, good. Good. I think that's good for Unit 5. There's nothing here that's just eating away at me to talk about, really. There's non-disjunction, but we kind of covered it already, so that's good. I do want to talk about purpose of mitosis is to grow and develop cells. It is to create identical daughter cells. And then meiosis is to create sex cells. Let's talk about regulatory sequences. This is kind of complicated for some folks. Okay. You see transcription up there, and you're just like, I gotta see it. Okay, we'll just briefly do this. Just briefly. Talked about this last night, so I'm happy to do it again. But this is a great picture. Look at this picture. You start with, where'd it go? Start with DNA, and transcription is going from DNA to RNA, just making a complementary RNA strand from the template strand of the DNA. And translation is making a polypeptide from the RNA. 5 FRQs the whole year. Man, I'm sorry. I really am sorry. I bet my students saw, I don't know if any of my students are in there. I saw one earlier. If any of my students, I bet we saw, counting the review weeks, I bet we saw 40 FRQs. I bet we saw 40. Yes, epistasis. Yeah, we're getting ready to talk about epigenetics and, yeah. Okay. So, lack operon. First of all, operons, yeah, I don't mind going over the codon chart. Actually, I can do that. Yeah, we reviewed for 3 weeks. I actually showed my students that schedule on the first day of school. Told them they'd have 3 weeks to review, and then we did. Because I don't play. Here's a codon chart. It's just literally first, second, third base, and it is a codon. Yeah, the hydroxyl group and all that stuff, not very important at all, really. Yeah, I'm sorry for some of your experiences. Really, I don't know what to say. I really do feel bad for you. I plan out my entire year before I leave this year, actually, and I stick to it. One of the first things I tell my students on day one is, you might get behind, but I won't. So we go fast. We actually have a lot of breaks too. We just kind of chill. It's a nice chill year. Yeah, we can look at phylogenetic trees. All right, so I'm gonna go over regulatory sequences real quick. Getting off track here. So regulatory, an operon is a regulatory sequence for prokaryotes only. Right, it's a very simple system. You have like Z, like Y, like A. These are examples, right? You don't have to know this specific one, though it is very common. These are genes that are being transcribed, and they're only transcribed in particular times, right? Most of the time, the repressor is going to be sitting right down on top of the operator. I mean, just look at these words. They tell you what they're doing. You probably need to know about them, but it's going to be pretty edge case. It'll probably be in the prompt. The repressor, what is it doing? It's repressing things. What is the operator doing? It's operating things. What is the promoter doing? It promotes this whole business, right? It's like, this is simple. Don't let these words bother you. RNA polymerase, what does it make? It makes the RNA polymer, right? This isn't bad. This isn't hard. Just look at the words and let them comfort you. These words are comfortable words, right? And so RNA polymerase wants to make the RNA, right? It wants to transcribe these genes, but it can't because the repressor's in the way. If we could just get rid of that repressor, how do we get rid of the repressor? Well, we're going to have this little molecule right here. It's going to induce this. Sometimes this is called an inducer. And when it binds the repressor, it lifts off, then it's able to transcribe those genes, right? Capsite, not important for beyond the scope of this class. This is just this picture. But the capsite is probably it's probably just some little sequence at the beginning of the gene to sort of set it off. So back with regulatory sequences, cell differentiation by definition is the regulation of genes, right? How is that? So let's take like a thyroid cell, right? A thyroid cell knows how to be a thyroid cell. It has all the information to be a lung cell or a pancreatic cell or a smooth muscle. It's just not doing it. Why is it not doing it? It's because those genes are being regulated and the thyroid cell genes are being expressed. So there has to be something that is turning those other genes off and allowing these other ones to be on. It's sort of like, it's like the restricted section of a library, like Harry Potter, if you're into that. In the thyroid cell, all the other areas are restricted except for the thyroid cell section. It's not restricted, right? So how are those genes regulated? There's a lot of ways. Some of them are transcription factors. These transcription factors are genes that will either allow for transcription or block transcription, right? There are some small RNA molecules that can actually prevent transcription or prevent translation, right? Just depending on the prompt. So that you don't have to know all of this stuff right here. Lord, I don't even know what any of that stuff is. You don't have to know it. Specifics are not important. You just have to be able to read a prompt and understand. And when you see things like translational repression, you're not afraid because you know what those words mean, right? That is this whole test, is just being able to look at some words that you're familiar with and not be afraid of them, right? And then just read the rest. We haven't talked about mutations. I'm going to skip past mutations. They're not too hard. I was going to look at epigenetics. I don't have epigenetics on here, unfortunately. Yes, small RNA can be positive. It can be a beneficial thing because there can be a reason to prevent transcription and translation. So I don't have an epigenetics slide. I do. It's just not here. It is going to be he. And it's probably going to be... really? Maybe it's this one? Maybe it is this one? Wow. Here we go. Here's epigenetics. Epi means on, right? Like an epidermis. Yeah, we're going to do cladograms. Epi is like on, right? So like epidermis is on the dermis. And so anything that's on the gene, not the actual A's, T's, C's, and G's, but is on the gene from the outside that affects transcription is considered an epigenetic, right? Could be reversible. Like histones are a great example of this. DNA can't be transcribed when it's bound up on we're going to do an FRQ review for sure. It can't be bound up when it's on the histone, right? So something has to come and release it. Or methyl group. A methyl group is a nonpolar thing that prevents DNA from being transcribed. And so anytime you see this word, just know that it's something, it's methylation. Anytime you see this word, just know that it's something coming from the outside that's affecting the DNA molecule from the outside and preventing transcription, typically preventing transcription, which is then going to prevent translation and ultimately protein synthesis. Yes. So I'm going to do, what was I going to do? Oh, yeah. I have to go here for that. Well, wait, where is it? Here we go. Let's see. Phylogeny. Okay. So let's look at this. All right, perfect. Cladograms and whatnot. So this is a phylogenetic tree. What is the difference between it and a cladogram? Phylogenetic tree is going to show time. Literally, time is, like these two, Vampirus and Electo, happened sooner than Meleta and Homo sapiens, right? Right. So that's the idea. It's just time. And so, one of the exam that we had last year, the 24 FRQ, actually had you draw, not the practice exams. 2024 FRQ had you draw on a phylogenetic tree, right? And it had you basically differentiate when did the pluses and the minuses differentiate from one another. Well, they differentiated from one another right here at this X, right? Because at this X, the pluses happen and then the minuses happen, right? And so that's the idea there. And so cladograms are a little more specific because cladograms are based on, yes, correct. That is the way that you can understand it. Some species are sooner than others. So, yeah. So cladograms also show time. Time is here, and then time is here, whereas the oldest thing is down here, old, right? And the more recent things are like out. For that one FRQ, the pluses and minuses just had to do with a particular trait. Yeah, real Lankin, we actually went over pedigrees just a few minutes ago. So if you can, I don't know if you can rewind the stream or whatever, but we went over those. Yeah. The important thing for cladograms is the derived traits, right? They're always going to have these traits associated with them. Like here is going to be a trait. In this case, it's jaws, right? And so down here is some sort of how do you know the outgroup? Okay, good question. I'll get there. Down here is going to be some sort of organism that represents the first jawed animal. Everything in this circle, wait, let me get a circle. Everything in this circle, right, has jaws, right? Because it's all going forward out from that point. Behind it doesn't have jaws. And so that's where the term cladogram comes from, is this is a clade. You don't have to know that, but that's what it's called. What about here? This is the ancient tetrapod here, right? All of these things are tetrapods. They're also jaws, right? Because jaws is going this way. They're also vertebrates because it's going this way. But backwards, right? Backwards, this fish is not a tetrapod, right? And so how you can use a cladogram is ways that I see them used is they may ask you which is more closely related to the bear, the chimp, or the lizard. And it's the chimp, right? Because they have, you can literally just count 1, 2, 3, or 1, 2, 3, 4. Literally, it's just like Monopoly. You just count. There's how close they are together, right? And so the outgroup is going to be the group on your cladogram that has the least number of derived characteristics, which is what I have over here. Oh, look, my definition is pretty close to what I just said. I'm pretty consistent with myself most of the time. Sometimes your cladogram will have an outgroup that has nothing, like no characteristics, and sometimes it'll have like whatever one, like the lamprey here is the outgroup. And what is the purpose of the outgroup? The outgroup is a basis of comparison, right? It serves as a basis for comparison for you. Oh yeah, and then sometimes you'll see this is not that important, but I have seen this though, so let's just look at it briefly. If you were given a table like this and you were asked to construct a cladogram, you probably won't be asked to construct a cladogram though. You'll probably be asked to like figure out where something would go, right? And so how do I construct it? Well, I can look at the thing that has the least number of derived characteristics. That's fish, right? And I'm going to draw my cladogram like this. And everybody's finding their, I mean, this is the place to be, right? Y'all, you need to go invite all your other classmates, especially you guys who only saw like 5 FRQs. This is just horrible. I'm sorry. This is bad. It's just bad. Endocrine, paracrine, autocrine signaling could be on the test. It could be auto means self, right? Para means like regional, and endo means like inside. And so I think of it's like in the blood. And so endocrine is like a long distance, paracrine is regional, autocrine is self. So fish has the least number, and so fish is going to go up here, right? And then what is the characteristic that fish have? They have a vertebra, and so you write it right here, right? And I'm not going to do this whole thing just for sake of time because I want to go over the FRQ too. And then you just keep going, right? And you'll see maybe there's like lizards and birds have the same. And so they're going to be closely related to each other, but not related to gorillas and rodents because, well, gorillas and rodents don't have eggshells. And so you'll have like lizards and birds kind of doing that sort of thing. I think juxtacrine is touching, but that's a word that you don't see as often. Because I think paracrine can be used in that same way. And the reason I know juxtacrine is because if you think of juxtaposing two things, you're putting them side by side because the word it literally means side. Yes, I will do that. Let's do a Hardy-Weinberg together. Let's do a Hardy-Weinberg. I'm actually just going to come up with it off the top of my head. Oh, here's one. Whoops, I inadvertently, oh no. I inadvertently zoomed my chat, and I don't know how I did it. Whoa, no. Oh, there it is. Okay, here's Hardy-Weinberg. This is a problem that I came up with in class. All right. Perfect. So in a population of 1,000 bears, I have a lot of bear illustrations, by the way. So you have 117 that have no ears. This is a recessive condition. How many bears, notice this is how many bears. So it's going to be a number from 1 to 1,000. How many bears are homozygous dominant for ear presence? All right, so let's walk through this one. All right, a couple things that we know concerning this. I'm just going to write them here. We know that P is equal to, I'm going to convince this to stop auto-capitalizing for me. Please stop. Is the percentage to, or is equal to the percentage of big A, and we know that Q is equal to the percentage of little a, right? And because of that, we could derive that Q squared is equal to the percentage of little a, and 2pq is equal to the percentage of big A little a, or heterozygous, and then p squared is equal to the percentage of homozygous dominant, right? And we know that p plus q equals 1, and p squared plus 2pq plus q squared is also equal to 1. All right, we know that those are all true. All right, so what I tell my students is always solve for q squared first. Why do you solve for q squared? Why do you solve for q squared? Because there are 883 bears that have ears. Those 883 could be big A, which is p squared, or big A, which is 2pq, right? And so then you don't want two variables. But little a is always q squared. So I know that q squared is equal to the percentage of little a, which is 117 over 1,000, right? Which is equal to that, right? And we're going to go ahead and round it up to 0.12. And so if q squared is equal to 0.12, then q is equal to, let me pull up my handy dandy calculator. 0.12 is the square root of that. 0.346, or we're just going to say 0.35. A lot of rounding, that's okay. And if that is equal to, if q is equal to 0.35, then p is equal to 0.65. And I'm asked to say how many bears are homozygous dominant, which is going to be the percentage of this, right? And so that's going to be 0.65. Actually, I'll just do it on the screen so you can see. That's going to be p squared is equal to 0.65 times 0.65, and that's going to equal that, which is going to round up, or times 1,000, because that's the number of bears that we have, times 1,000, and that's going to equal 422. We're going to round it to 423 bears. All right. That's my answer. So I rounded a couple times, and so there could be a pretty good range there. I rounded a couple times where I probably shouldn't have, and yeah, that's probably why there's a little bit of a range. Correct. So anytime there's some sort of figuring like this, it is going to, AP will give you a range there. So. All right, so that's Hardy-Weinberg. What else was I asked? Chi-square. Yeah, I guess we can do a chi-square question real quick. We'll just do it down here. Let's do a chi-square problem real quick. We'll say that. How do you do chi-square and Hardy-Weinberg in genetics? All right, let's do that. Let's do that. There's a problem on one of the practice exams I can't show you, but it's similar to this. So let's look. Let's look at it. So let's say we have a population of mice, and in mice, this population is there's 100 mice, and 75 of them are big A, little a. 15 of them are big A, little a. And 10 of them are little a, little a. right? And so if this population is at Hardy-Weinberg equilibrium, then if these are the observed values, and then we could say, is this population at Hardy-Weinberg equilibrium, right? And this is a population of 100 bears. This is like a 5-step problem, right? So this is similar to this is a pretty famous question from one of the practice exams. So how do you figure this out? Well, there's 200 organisms. You need to know both of those, yes. 200 organisms, or 100 organisms, 200 alleles, right? I may have missed part of this problem, so it may not turn out right, but we'll go through it anyway. We'll see if it's going to turn out. If there's 200, right, then how many big A's are there, right? There's 150 plus, there's 150 because there's 75 big A's times 2, right? And then 15 more because there's some of these here. So the percentage, P is equal to the percentage of big A, which is equal to 165 over 200, which is equal to that, right? Right? Is everybody tracking with me? And if P is equal to 0.825, then Q is equal to 1 minus P, which is equal to 0.175, right? All right. How are we doing on this so far? Pretty good. Pretty good. And now let's see if this population is at Hardy-Weinberg equilibrium. How do we get 165? The percentage of big A, right? So there's 100 individuals. Each individual has 2 alleles, right? Each individual has 2 alleles. So 100 times 2 is 200. How many big A's are there in this population? Well, there's 75 times 2, right? Because there's 1, 2 here times 75, so it's 150 plus 15 out of 200. So that's where we got that. Okay. And so P squared is 0.825 times 0.825 equals that. So that's P squared, right? And that should be big A should equal 68, right? Right? And 2PQ, 2 times 0.825 times 0.175 is equal to 0.288, which is equal to big A little a, which is equal to is equal to 29 mice, right? And so that means little a is going to be equal to those two numbers added together minus 100 or subtracted from 100, so it's going to be 3. So these are my expected values, right? And over here are my observed values. And now I can just do chi-square. So chi-square is going to be, sorry, you can't see my calculator, but it's going to be 68 minus 70 observed minus expected. So it's going to be 75 minus 68. Whoops. Squared over expected, which is 68. So it's going to be 0.72. Look, I'm showing y'all a super hard version of this, but it was just a way to kill two birds with one stone. A lot of teachers have no idea how to do this problem, so I'm not disparaging your teachers. I'm just in a Facebook group with them, and they don't know how to do this problem. So 0.29 is the expected number of big A, little a, but 15 is the observed, right? So 29 minus 15, and then squared, and divide that by expected. It's going to be 29, so it's going to be 6.76. You know, it's funny. That's a funny question, Kevin, asking, is this going to be a multiple choice question? This was a multiple choice question on one of the practice exams. This exact numbers and everything. So yeah, if you get this multiple choice question, by the way, skip it. Go back to it when you're done. Don't spend 10 minutes working on for 1 point. Don't do it. So it's just go back and do it. That's my advice. I don't know what that 0.2 that. Oh, this is just wrong right here. It should be 5 there. Sorry. That was just a mistype. And then the last one is, 10 observed minus expected, so minus 3 squared divided by expected, which is 3, and that's going to be 16.3, and that's going to be equal to, all right. You do need to know rRNA, barely. The other two things, probably not. So your chi-square equals chi-square, right? Do not take the square root of chi-square. And since I have 3 things, how many degrees of freedom is that going to be? If I have 3 things, right, you have 2 choices, right? And so 2 degrees of freedom. I'm going to go back up here and look at my FRQ sheet here, or my formula sheet, just so we can see the table together. I have 2 degrees of freedom. I'm going to use 0.05, so it's 5.99 is my critical value. My chi-square was way higher than that, and so you have to reject the null hypothesis. There's something going on that's fishy with those numbers. Okay. I'm going to spend the next little bit going over this FRQ here. And so we're going to keep all of our questions here on the FRQ. Yeah, no problem, Cujo. I do it for my students, and I'm glad that other people's students can get the benefit for them as well. So, and I don't get paid. I am not monetized on YouTube yet. You never know, maybe soon, but even then it's not going to be enough to feed my family or anything. So yeah, this is 2021. I am monetized on Twitch, by the way. If you're ever bored, you can just follow me on Twitch. If you want to watch some old man play Minecraft or Fortnite. There you go. Maybe tomorrow evening after the exam, we can all watch me play. This is my Twitch handle, by the way. You can come watch me play Fortnite or Minecraft or something like that, and tell me about the exam. Drown your sorrows watching an old man get beat up on Fortnite. Okay, so here let's do this. Sorry, got sidetracked on games. It has been a bit since I've looked at these FRQs, so I make no guarantees. But here we are. Thanks, Amalika. I appreciate that. Yeah, I'm in a 3-person Minecraft server right now. I can't imagine having 60 in there. Jeez, that would be crazy. Be absolutely bonkers. All the cows. Okay, sorry. Polycystic kidney disease as an inherited disease that causes water loss from the body and affects cell division in kidneys. Yeah, we can look at that. Okay. Water loss in the body and affects cell division in kidneys. Speaking of cell division, here we go. Because water movement across cell membranes is related to ion movement, scientists investigate the role of sodium-potassium ATPase, also known as sodium-potassium pumps, in this disease. Okay, actually, I'm just going to keep reading. Uabane, I guess that's how you pronounce that. A steroid hormone binds to sodium-potassium pumps in plasma membranes. Individuals with PKD have a genetic mutation that results in an increased binding of uabane to that. Scientists treated normal kidney cells and PKD with increasing concentrations of uabane and measured the number of cells and the activity of sodium-potassium pumps. After a period of time, the scientists hypothesized that a signal transduction pathway includes protein kinases MEK and ERK and may play a role in PKD symptoms. Okay, this is so I'm just going to stop here and I'm just going to highlight the fact that you do not need to know what polycystic kidney disease is, but there's a lot of stuff that we know. When you look at this problem, right? We know some of this stuff. We talked about pumps, right? We talked about plasma membranes. We talked about signal transduction pathway, right? There's stuff in here that we know. So grab a hold of the things that you know, ignore the things that you don't until you can't ignore them anymore. Oh, look. A fun little picture. It's like some graffiti or something. Anyway, this is signal transduction, right? Signal transduction response. Let's do this. Part A is always not related to the prompt. Describe the characteristics of the plasma membrane that prevent simple diffusion of sodium and potassium across the membrane. So why can't they go across the membrane? It's because of this. It's because they're charged. The characteristics of the plasma membrane are such that the plasma membrane has a polar outer and inner part and a non-polar center, and that does not allow the passage of charged particles. Explain why ATP is required for the activity of sodium-potassium pumps. Because they're pumps. ATP is required because it is pumped across the gradient, and that is a form of active transport. Right. Notice how I'm keeping my answers short. I'm not trying to write a book about these things. You don't need a book. You don't need a book. Identify the dependent variable in the experiment in figure 1. How do you know dependent variable? It's the y-axis, number of cells. And look, it's identify, and so I'm just writing number of cells and I'm walking away. I'm not writing the dependent variable in experiment 1 is number of cells. I'm just writing number of cells and I'm walking away. I'm not trying to write all that. I'm not doing it. Justify the use of normal human kidney cells as a control experiment. Okay, why? Because you need a basis of comparison. That is my reason. My justification is I need a basis of comparison, so I'm going to compare disease cells to normal cells. Justify the use of a range of uabane concentrations in the experiment represented in figure 1. Sample size, right? You want a lot of sample size in order to find out which of those concentrations is actually causing a change. So that's what you do. Good. We've already got through A and B. We've already answered, 5 of the how many ever points there are. Part C. Part C is usually where it starts to get in the nitty-gritty. Let's look. Based on the data shown in figure 2, describe the relationship between the concentration of uabane. Oh no, Let's look. Describe the relationship between the concentration of uobane and sodium potassium pump activity on normal cells and PKD cells. Scientists determined that activities. So what was the question? Describe the relationship between the concentration of that and activity. I would say that as concentration of uobane goes up, the activity goes down. That would be my description. Calculate the expected ATPase activity in PKT cells treated at 10.6. So let's see. So it's 30. There's some other thing here. Yeah, we'll do Calvin cycle. Remind me when I get through this. If we haven't talked about it yet, I'll do it real quick. So 10.6 is 30. And what was this? Something here. 150 units. Okay. So how many units per second is that? It's 150 times 30. So that's 4,500. All right. Part D. In a third experiment, and if I'm going too fast here, slow me down. But this isn't hard. You just have to look at each one separately. Don't look at this big block of text and be like, oh no, I'm cooked. You're not cooked. Every sentence is just by itself. In a third experiment, the scoring guideline says 40 and 50 on parts. So what did I do wrong? Who knows? I'm actually curious what I did wrong on that one. How did I get that wrong? Oh, I know what they did. They divided that by 60. Let's see. Nope, that's not what they did. I have no idea. I have no idea why I missed that one. I multiplied it by 30 because it was 30 here. Percent. Oh, 30%. 30% activity. Gosh, I have no idea. I'm at a loss on that one. I'm sorry. I would have to actually sit down with that part two and look at it. I don't know how they got that. That is going to be where I am definitely weak when it comes to this material. So perhaps there's someone in here that knows how that was solved and could solve that for us, and who knows more than I do. But I'd have to look at it in a little more depth. No, glad to help for sure. So you guys are asking about Calvin cycle and Krebs cycle. I went over it in pretty good detail last night, and so you should just go watch those. Seriously, I went over it and I was fairly thorough, and so just go check out those streams, and went over that fairly thoroughly last night. So yeah. Part D. In a third experiment, scientists added an inhibitor of phosphorylated MEK to PKD cells exposed to 10 to the fourth, based on Figure 3, predict the change in relative ratio of ERK to PERK. Oh. Yeah, if I'd have just changed my 30 to 0.3, I would have got it right. Yeah, that's what I did wrong. I multiplied it times 30 instead of 0.3. Thank you, Pancake Face Syrup. I needed to say your name. I just needed to. It was perfect. It's a great screen name, by the way. Pancake Face Syrup. Sorry, predict the change of relative ratio of ERK to PERK in urbane treated with an inhibitor compared without the inhibitor. Okay, so I'm actually looking at this picture here. So if there's an inhibitor added here, then this is not going to happen. And so predict the change, there's going to be less ERK to PERK. Why? Because the inhibitor is going to slow down this portion of transduction, right? If there's an inhibitor here, it's going to break this next portion of the process. Simple. Use the data in Figure 1 and signal transduction pathway represented to explain how the concentration of cyclin proteins may increase PKD cells with 10 to the 4th. Okay, PKD. Cyclin is this is more cells here, right? So the number of cells is significantly more at 10 to the 4th. Yeah, so why is cyclin increased? That is because the number of cells is increasing and there's more cell division. Therefore, there's more cyclin overall. Because the more cell division there's going to be, the more cyclin there's going to be. Cyclin is present during the cell cycle. So, yep. Good. Here's a nice pedigree for us. Geneticist investigated the mode of inheritance of a rare disorder that alters glucose metabolism and first shows symptoms in adulthood. Geneticists studied a family in which some of the individuals of generations 2 and 3 are known to have the disorder. Based on the pedigree, geneticists concluded that the disorder arose in individual 2 here and was caused by a mutation in mitochondrial DNA. Okay, perfect. Man, we're on top of this. On top of it, right? Mitochondrial DNA. We're familiar with this. So we're going to go. And then here's some things. Some nice numbers for us. Part A is never about the prompt. It always stands alone. So let's look. Disorder alters glucose metabolism. Describe the atoms and types of bonds in a glucose molecule. The atoms are carbon, hydrogen, oxygen. Types of bonds are covalent. Hey, we talked about that earlier. Yippee. Part B. Use a template in the space provided for your response. Construct an appropriately labeled graph. Okay, so let's look at this graph. You're going to have what are cyclin-dependent kinases. They're enzymes that bind to cyclin, and when they bind, they form something called maturation promotion factor. And as maturation promotion factor builds in concentration, it causes the cell to go into mitosis. And at metaphase, anaphase break, the cyclin and CDK, or the cyclin begins to break down and causes the cell to finish the cycle. So you're going to have a bar graph here. How do I know it's a bar graph? Because I have 8 different individuals and we're comparing them. And average blood glucose levels of individuals. So I'm measuring blood glucose levels, right? So they're going to be on the y-axis, and my individuals are going to be on the x-axis. So I'm going to have essentially 8 bars on this graph. Determine one individual who is both at risk of developing the disorder and has a significantly different level from 4-1. So the way that you can look at this, average blood glucose level, and the disorder is an abnormal. Okay. I haven't seen Boston whisker plots at all on an exam yet, so they could be. You're not going to be asked to draw one, but the way that you interpret it is quartiles. There's like inner quartiles and outer quartiles, but I wouldn't worry about it. It's edge case. Most of your graphs are going to be line graphs, bar graphs, and stuff that looks like this, right? Simple stuff. Keep it simple. Focus on the 80%, right? Instead of the 20. All the little edge cases. For some reason, teachers just love to talk about edge cases, and will spend like a week on them. I don't get it, but they do it. Oh, here's this. Okay, this is what I'm looking for. Okay, normal is, so at risk is here and affected is higher. So okay, good. Let's determine one individual who is both at risk and has a significant. So at risk is going to be between 140 and is significantly different than this one, 140 to 200. Okay. So if you had a graph, it would be much easier because you could see the error bars, right? I'm not even going to answer this question because it's just going to take time to do the math, and I'd rather you see, but I'm not going to do the graph for you. But you're basically, which one is not overlapping with 4-1 and is in this at-risk range here. Based on the pedigree, identify all the individuals in generation 4 who can pass their mutation. Okay, good. If it's mitochondrial DNA, who passes it? The mothers pass it. So we're going to go individuals 1, 2. Wait. Yep. 1, 2, 4. 5 can't pass it, right? Because 5 didn't get it. 1, 2, 4, and that's it. You can get a 3. You can do that for sure. Based on the fact that individual 2 is affected, the student claims that the disorder is inherited in an X-linked pattern. Based on the student's claim, predict which individual of generation 3 will become or will be affected by the disorder. So 2, 2 is affected. A student claims that the disorder is inherited as an X-linked recessive. Based on the claim, predict which of the students which of the individuals in generation 3 will become affected. So if 2, right, 2 has it and it's X-linked recessive, which individuals will get it? Well, if mom has it and it's X-linked recessive, mom only has recessive on her X chromosomes. Dad only has dominant, and so mom gives her X chromosomes to sons, and so only her sons are going to have it. So individuals 4 and 8, based on that. That's my prediction. Justify why the data do not support the student's claim. The data do not support the student's claim because the daughters also have it, and they can't possibly have it because dad doesn't have it. And so there we go. That wasn't too bad. On to the short ones. Number 3. I like these. I like the short ones better. Don't pay attention to the deviation or any of that stuff. you're just going to do 170, and you're going to go plus 15 and minus 15. So you're going to go 170, and then you're going to go up to 85 and down to 55, right? That's it. All of this stuff is like if your teacher spent 2 weeks going over standard deviation, I'm sorry. It's just not on the exam, ever. Ever. Question 3. Researchers hypothesized that the plant compound resveratrol improves mitochondrial function. To test this hypothesis, researchers dissolve res in dimethyl sulfoxide, DMSO. The solution readily passes through cell membranes. They add the chemical to mammalian muscle cells growing in a nutrient-rich solution that contains glucose. They measure ATP production for several time points after an addition of that chemical and find an increase in ATP. Okay. No, it's always going to be given. You don't have to calculate that garbage at all, ever. I don't know why the formulas are there. It's not even in our standard. Like you don't have to calculate. Yeah, I know teachers spend a ton of time on it. I don't know why. So, you do have to use it, but you only have to use it to tell if the error bars are overlapping or not. That's the only time I've seen it. You guys might get thrown some kind of crazy curveball, and if you do, I'm sorry. But I've been teaching this class for a long time, and I've not seen it yet. So, describe the primary advantage from mammalian muscle cells in using aerobic respiration over fermentation. Muscle cells require a lot of oxygen, and aerobic respiration uses oxygen. That's it. Identify an appropriate negative control for this experiment that would allow the researchers to conclude that ATP is produced in response to that treatment. The appropriate negative control would be cells not treated with the chemical. It's going to get changed just a little bit, but it's not in substance. It's mostly, if you can imagine, I'm a weird illustration person, and I use books a lot, so I'm just sorry. But if you can imagine like a bookshelf, right, and that's the AP Bio curriculum, they're just going to be rearranging the books. They're not taking anything off or adding anything to them. They're going to arrange them in a way that makes more sense. That's the change. The actual content is not going to be changed any. I'll have to change my stuff a little bit, but not in substance. Aerobic respiration generates a ton more ATP, yes. We didn't do a multiple choice review because all the multiple choice questions are gated. If you want a multiple choice review and your teacher has not given you access to AP Classroom, then I highly recommend Khan. No formulas that you need to memorize. They will give them all to you. Part C. Predict the effect of short-term ATP production when resveratrol treated mammalian muscle cells are grown in a culture medium that lacks glucose. The short-term effect would be a decrease in ATP production. Why? You don't need to say why because it just says predict. Don't need to say why. Don't tell them why if they don't ask. The researchers find that resveratrol what's so bad? Stimulates the production of components of the electron transport chain. Oh. Researchers claim that treatment of that will also increase oxygen consumption by the cells, and glucose is not limited. Justify the claim. Why is oxygen going to be consumed more if the ETC is being produced more? Because oxygen is the final electron acceptor on the ETC. It's being consumed. It's made into water. That's my justification. Yeah, it's going to be online on Blue Book. Speaking of Blue Book, if you're taking it on your Chromebook tomorrow, or whatever, and you have your school Chromebook and your teacher hasn't had you log on already, you should try to log on to Blue Book and make sure it works fine. What was A? Muscle cells need a lot of oxygen. That's all right. You have time to reread it 2 or 3 times. You have time, because you're not going to write 10 paragraphs for each one. You're going to write like a sentence, right? You're going to have time to just read and chill. You're not going to write like a book. Don't write a book. It's science teachers grading it. They don't want to read your book. They want to read a sentence. Number 4. In 1981, a single immature male Geospiza canerstros, man, I love these words, finch, flew more than 100 kilometers from the Galapagos Islands of Hispaniola to the Galapagos Islands, Daphne Major. Of the Noti, I don't know what that is. I mean, I have videos answering 2024 and 2023. I don't know if that's what you're asking or not. Yeah, only write down what you need. I'm assuming that I'm assuming your school will provide a Chromebook for you. I'm really sorry that you haven't been told that, or you've been told that and you probably weren't paying attention. Our schools are 1-to-1, so our students will have a Chromebook. But the students that forgot their Chromebook will also have a Chromebook, so you'll probably have one. The immigrant finch bred with a female, a species finch common to Daphne major, F1 finches. Oh, look, here's some Unit 5 stuff. The F1 finches and later generations interbred only with their lineage. By 2012, scientists counted 23 individuals, including 8 breeding pairs, within this hybrid lineage of Daphne major. The hybrid lineage became known as Big Bird. Big Bird. Birds with different beak shapes and sizes eat different types of food. The dimension of Big Bird beaks. Man, I love Big Bird beaks. That'd be a fun band name. Relative to the beaks of the major competitor finches in Daphne major are shown. So you have Big Bird here. So this is a great example of a scatter plot graph, right? A scatter plot is plots between 2 seemingly unrelated variables. Depth and length aren't necessarily related, and so you have all of these little plots here, right? And then you have Meiosis here that wasn't even mentioned. And then you have Fortis and you have Scandens. Wait, no. I don't know. Fortis. All right. The Big Bird lineage became reproductively isolated from Fortis. Describe one pre-zygotic mechanism that likely contributed to the reproductive isolation of Big Bird lineage. Big birds have bigger beaks, right? And so here's the Fortis beak, which is smaller than Big Bird beak. And so they have to eat different things. And so I would say that the pre-zygotic mechanism would be habitat isolation. They were in different habitats because they eat different things. That is strengthened by this sentence here. Based on the figure and data 1, explain why Big Bird population has been able to survive and reproduce on Daphne major. Why have they been able to survive? Because they are eating something different than the competitor. So they're eating their own niche, right? They're not competing for a niche. They're in their own niche right here. And so that's why they've been able to survive and reproduce. Virus infects and kills all Meiosis on Daphne major, but does not affect the other finches. Assuming food type and availability stay the same, predict the most likely change in beak phenotype of big bird population after 6 or more generations. So you'd expect then, because this niche is, all these are dead, and so since that niche is available, you'd expect that big bird may undergo like a directional change, right, to where it has bigger beaks over time in order to exploit this new food resource. Provide my justification. My justification is because there's a new food resource available, and that would be my justification. Number 5. Annual plants complete their life cycle, including germination, seed production, and death within 1 year. Ambrosia trifida, giant ragweed, is an annual plant that readily colonizes any land that has had a disturbance such as plowing. Interesting. The plant is considered an invasive species in regions outside its native range. In a particular region, the seeds of A. trifida germinate from early March to the end of summer, while the other seeds on the annual plants require warmer soil temperatures and thus germinate from late April through end of summer. Researchers studied the influence of A. trifida on biodiversity of other plant species that grow in the same field. In early spring, researchers marked off identical plots in land and plowed previous fall and not replanted with new crops. All plants that grew on one half of the plots were left untouched, while all germinating trifida seedlings were removed from other half of the plots throughout the spring and summer. In the late summer, researchers counted and identified all plants that grew in the plots. Distribution of plants is represented here. So you have trifida, okay, so that's kind of crazy reading. So what is going on here? Basically, A is left untouched. All the trifida was removed from B, right? And so before I even go look at the problems, what do I notice? A has very little diversity because it's trifida is there. B has 3 different kinds of plants there because there's no trifida. Just saying that out loud because we're going to have to say that very same thing again, I guarantee you. Describe the cause of logistic growth in ragweed population. A cause of logistic growth of the ragweed population. So logistic growth has to do with growth in relation to carrying capacity, right? And so the logistic growth would be any sort of limiting factor, right? And so you could say the availability of warmer soil temperatures as a limiting factor. Or you could say seasonal. You could say there's a couple things that you could list there. Any sort of limiting factor is going to be right there because logistic growth has to do with growth based on a carrying capacity. Based on the representation in Figure 1, explain why scientists claim that Plot B would be more resilient than Plot A. Yeah. Vocab for Unit 7 and Unit 8, they're both pretty vocab rich. I actually have 2 videos. I have a Unit 7 comprehensive and Unit 8 comprehensive. Consequently, they are my most watched videos over the last month. And it's a lot of the reason is because a lot of people's teachers don't get to those 2 units. So I know some teachers just do Unit 8 as like a take-home or something. So sorry. I don't know why that happens, but it does. So yeah, encourage you to watch those videos. Watch them on 2X and just get that vocab in. Get it in. It's a lot, but it's not that bad. It's probably stuff you've had since middle school. Yeah, I'm starting to wear out. I'm starting to wear out a little bit. You're right. I'm sorry. I'm gonna get it. I'm gonna like pep up, and I'm gonna get this last little bit done. But yeah, I've got about probably 15 minutes left in me. So let's do this. Resilience. It's more resilient because it's got more biodiversity, right? We just said that before we went down here and looked at the problems. Number C. In a third group, plots, researchers removed all seedlings from other plants that germinated before June 1. All plants that germinated after June 1 were left untouched. Using the template in the space provided for a response, represent the expected plant species that would be found in this third group of plots 3 months later. Draw no more than 12 samples. Assume all other environmental conditions are the same. Okay, so we're going to be given you're going to be given a square, right? You'll be given a square to draw in. I don't see it on here, but you will be given a square. And I would say that it's going to look similar to this one. I would say that it's going to look similar to this one because removed all seedlings of all the plants that germinated before June 1. All plants that germinated after June 1 were left untouched. I think that it's going to look just like this one. That may be wrong, but that's what I think. Because it says through the end of the summer, which is September. So, explain how invasive species such as ragweed affects the ecosystem diversity. Ragweed is going to disrupt ecosystem balance because it is going to require resources, right? And it also doesn't have any natural predators. It doesn't have any natural predators or natural diseases or anything like that that are going to be limiting factors for it. And so it's going to come into an ecosystem where it has not evolved and other organisms have not evolved with it. And therefore it's going to disrupt the normal balance of that ecosystem. Question number 6. The small invertebrate krill species is adopted to cold seawater. Over the past 10 years, there's been a gradual increase in the water temperature of krill's habitat. Sustained increase in water temperature may ultimately affect the ability of krill to survive. One effect of higher temperatures is protein misfolding within cells. Krill have several HSP genes that code for heat shock proteins. These proteins help prevent protein misfolding and help refold proteins in their normal shapes. Scientists conducted experiments on T. intermis to detect changes in the expression of HSP genes when the krill were exposed to temperatures above 4 degrees Celsius. Experimental group of krill was maintained in tanks with 4 degrees Celsius seawater, then placed into tanks with 10 degrees for approximately 3 hours. Then krill were given a 6-hour recovery period in that 4-degree tank. Control group was moved from a tank of 4 degree to a tank of 4 degree with approximately 3 hours, then returned to the original tank. Scientists analyzed HSP expression by measuring the concentrations of 3 mRNAs, transcribed certain lots of stuff there. Okay. Identify HSP mRNA that has the slowest rate of concentration increase in the response. Okay. So average mRNA concentration. And this says which of the mRNA has the slowest rate. It's going to be 3, right? Or wait, in response to the heat shock, which is going to be after the heat shock. It's going to be 3. Oh no, no! This one goes down, so it's going to be this one's kind of wonky, isn't it? I'm still going to go with 3. No, it's going to be square. It's going to be 2 because it literally has a negative rate. So it's 2. Sorry. Describe the trend in the average concentration of mRNA-1 throughout the experiment. The trend is to continue to increase in concentration. Yeah, I mean they're on purpose. They're trying to sort of like trip you up. Sometimes they go too far and they actually have to go back and require 2 answers. There was literally one last year or 2 years ago where they had to allow 2 answers because the wording was so vague. Part C, scientists hypothesize that heat shock proteins translated from mRNA-1 play a greater role in the refolding of proteins than does mRNA-2. Use the data to support this hypothesis. Sure, because mRNA-2 goes down after heat shock, whereas mRNA-1 increases concentration after heat shock. So that's why. mRNAs 1 and 2 are transcribed from the same gene. Explain how a cell can produce 2 different mRNAs from the same gene. This would be alternative splicing, right? The introns are removed and the exons can be rearranged. Yeah, I wish they could be clear too. The reason that they try to like skirt the edges of vagueness is because they don't want anybody to score perfect. And that's not mean. You know, every plane needs a place to land. And if you score perfect, then you weren't properly assessed. That's kind of the way I think about it. So it's not a bad thing. It's just those questions stick out in our minds, and we don't like them. But it's such a small portion of the test. That is all of the 2021 FRQs. And so I will take just a few more questions. I am like starting to wane quite a bit, starting to get tired. And so, but if you have, I think let's just maybe limit our questions to like test day questions. If you have any questions about test day or what, kind of expectations, if you have those. Yeah, that's a good question, Outlaw. I think it's probably about 70% content, about 30% logic. And so if you have a pretty good grasp on your content, you're going to do better. But there are a lot of questions that you can sort of logic your way through. So yeah, that's a good question. Here's the exam breakdown. I mean, if I wasn't tired and I had a few more minutes to watch something tonight, I would watch a video on Unit 3, I would watch a video on Unit 6, and I would watch a video on Unit 7. Yeah. I actually have 3 Unit 3 videos. I don't know why I didn't make it into one video, but I have an enzyme video, a photosynthesis video, and a cellular respiration video. And then I have a Unit 6 comprehensive and a Unit 7 comprehensive video. Should you take the rest of the day off? I would definitely plan on doing nothing after your test. So, yeah, Cujo, for sure. You will have to make one graph, and it will be question number 2. Most likely. You'll have to make one graph. It'll be question number 2. And just to show you, too, like here's 2023. Just so you can, I don't know if you knew this or not, but what your teachers told you. So this is question 2. This question is, you can tell how many points each question's worth just by looking at the bolded letters. 1, 2, 3, 4, 5, 6 points, except for this word. Anytime you see the word construct, it's worth 3 points because that's the graft. All the other words are worth 1 point each. Yep, you're welcome. I'm really glad to do this. I love doing this, so I'm glad to help for sure. Yeah, question 2 is always a graph. I haven't ever seen it be question 1. Yeah, I mean, we do a lot of notes in my class too. I think explanations are really what make it, being able to explain your content and being able to figure out where a student is like struggling. Yes, you always modify a picture on an FRQ, and it's literally always question 5 every single time. Look, here's question 5, and it wants you to modify this picture. Always question 5, and it's usually going to be the keyword represent. Sorry, I haven't answered your question above. Please remind me of what your question is, and I'll attempt to answer it. Sorry about that. You are very welcome, Amalika. Always a pleasure. It would only be 2 points if it was 2 bolded words. That's it. So every bold word is worth 1 point, except for the word construct. It's worth 3 points. Yep, glad to help you, Cujo. Come watch me play Fortnite tomorrow night. Great question. I would watch the review videos. Do not go back through and watch 1.1 through 8.8 because that's going to take all night. Just watch the review videos, and they're going to be good for you. Man, you guys have made my evening. I don't know where y'all are from, but I'm sure you're from all over, and this has been a good time. Thank y'all for coming out. If you missed the stream, you can watch it. You can watch the one from last night also. And just so you all know, in a couple days, when the FRQs are released, I will make videos. I will make 6 videos. I'll make a video for each FRQ of me answering the FRQs. So you can watch and see how you did, or see where I'm wrong, because, who knows. San Jose, awesome. I would watch Units 3. If I was at 8:53 the night before the test, I'd watch Units 3, 6, and 7. That's what I would do. Yep, absolutely. Thank you guys very much, and good luck on the exam. Please, yeah, you can. That'd be great. You just go to my Twitch and sub to my Twitch, right? You can just go and sub to Twitch. Speaking of Twitch, weird plug here, but if you like watching Twitch and you want to watch an old man play video games, go there and watch. That'd be a fun. Come and chat with me while I suck it at Fortnite. That'd be hilarious. So yeah, but I will be answering the FRQs, and tell me how you did. Leave me a comment on this video or just any video. Let me know how you think you did on the test. I'd love to hear from you. Thank you all for watching.